Hello. So we're learning about life science and cell biology in a nutshell. So the nut just keeps getting bigger and bigger as we stuff more information in it. So we're learning about molecules and how they function within cells and that the kinds of molecules that exist and their functional groups. So functional group is a crucial part of the molecule which determines how it functions. So, similar functional groups have similar reactivity and affect the molecule in certain ways, making them polar or and making them interact with other molecules in specific ways which affect the functions of cells um, and cell signaling and how cells lead molecules go through them or um, participate in various reactions. Earlier, we learned about amino acids and how they make up chains of polypeptides, or aka protein molecules, which are translated off of mRNA molecules, which come off DNA um, genes and so genes in this way are able to code for certain proteins which are made in a cell, and usually this is done in ribosomes, the protein synthesis. So here we have a diagram showing the various amino acids which make up proteins, just to be a little familiar. So So glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, proline, serine, threonine, cysteine, tyrosine, asparagine, and glutamine. So all the amino acids have a basic uh, two carbons and an NH3. So hydrogen, a double bound oxygen, and a single bound oxygen. So you have that is the basic part of all amino acids that is in common, and as well the different unique parts highlighted here which make the amino acids different from one another. So glycine has the hydrogen of the carbon, central carbon. Alanine has a CH3 group. Valine has CH and two CH3 groups. Leucine has CH2, CH, and two CH3s. Isoleucine, CH, CH3, CH2, CH3. Methionine has two CH2s, a sulfur, and a CH3. Phenylalanine has CH2 and a 6 carbon alternating double bonds. Tryptophan, CH2, um, 4 carbons, and a nitrogen, NH, um, and alternating double bonds ring, and another 6 carbon with alternating double bonds. It's tryptophan, and then proline has CH2s, 2 of them, no, 3 of them, okay, in like a triangular shape ring structure. Serine, so those are actually non-polar, those side chains, 1, 2, 3, 4, 5, 6, 7, 8, 9 of them in those amino acids. And then these ones are polar side chains, so a CH2 and an OH. In serine, because of the OH or hydroxyl group, they're polar. So then in threonine, CH, CH3, and OH. Cysteine, CH2, and a sulfur, hydrogen, so cysteine, that's sulfur. Tyrosine has CH2, a 6-carbon alternating double bonds, ring, and an OH group. Tyrosine asparagine, CH2, carbon double bound oxygen, and NH2 group. So it contains nitrogen. And a glutamine also has nitrogen, so it's 2 CH2 groups, carbon double bound oxygen, and NH2. So those were 15 amino acids total, nonpolar and polar side chains. And then we have 5 more here, making 20 total amino acids. These are electrically charged side chains. In aspartate, you have CH2 and C double bound O and another O. So in glutamate, you have 2 CH2s and C double bound O and an O. Lysine, you have 4 CH2 groups and then an NH3 group, so nitrogen. And arginine, you have 3 CH2 groups and NH, and then a carbon bound to NH2 and then 2 NH2s, actually. Oh, this one's double bound and one is single bound, NH2. And histidine, CH2 to an NH. It's like a ring, 1, 2, 3, 4, 5. Ring NH2. I mean, 2 NH groups, an alternating double bond. So 20 amino acids total. Amino acids are comprised of a backbone consisting of a carboxylic acid, so COOH, which is and an amine group, NH2, linked by a carbon atom. So that's the backbone. The linking carbon atom is also attached to a lone hydrogen and the functional group of the amino acid coming off of it. The functional group of the amino acids provide the amino acids a set of properties, whether it is charged, polar, aromatic, meaning ringed, aliphatic, etc. So it's charged properties, and it can be small as a hydrogen atom, for example in glycine, or it can be complicated in a large ring structure like tyrosine, for example, or even tryptophan, which has 2 rings. So the functional group is abbreviated with R. So it indicates the location of the group on the amino acid. So NH3 and is the amino group, carboxyl COOH group, bonded to alpha carbon. At 7.2 pH, the carboxyl becomes deprotonated to carboxylate, COO minus. So it loses the hydrogen. So amino acids are in this way. So at biological pH, they lose their hydrogen and are just COO minus. So these R groups exhibit functionalities which are specific to the various amino acids, and the amino acids link up together to form polypeptide or protein chains. The one amino acid links up to another via their amino group of one binding to the carboxyl group of another, forming a peptide bond and creating amino acid residues and polypeptide chain. So this is the amino end and that's the carboxyl end. Functional groups are all the R groups, so specific. They may be polar